Welcome to Ask the Expert. Today we have Dr. Ping Wang, who also goes by Peter Wang. He's going to be talking about trends of nanotechnology and type 1 diabetes treatment. He is an assistant professor in the Department of Radiology and Precision Health program at Michigan State University. And before joining Michigan State University in 2018, he was an instructor at Harvard Medical School from 2014 to 2017 after he obtained his postdoctoral training from Athenula Martinez Center for Biomedical Imaging at Mass General. And his research focuses on molecular imaging and theranostic on cell transplantation and nanodrug delivery for type 1 diabetes and development of novel imaging techniques for cell-based therapy. He's a member of the Academy for Radiology and Biomedical Imaging Research, CECI squared, 2022, and SciLog, which is an Advancing Bioimaging Fellow of 2021. I'm going to put his recent paper in the chat, on his bio, in our chat for those of you who have interest in it. And welcome, Dr. Wang. Thank you for joining us. And I hope you can, letting someone else in here. I hope you can share your slides with us.
Yes, thank you, Dr. Wesley. Thank you for the invitation and for the introduction. So I'm gonna share my screen. Can you see my slides?
Yes. I don't, okay, perfect. Yeah, now it's perfect.
Great. Thank you for having me today. Before I start, I just want to say several words for this great platform. I have been participating in the science, sugar science since last year. Thank you, Dr. Wesley, for your dedication and contribution to this great platform, connecting researchers in the type 1 diabetes community from bench to the clinic. I personally enjoyed a lot and I have learned a lot. Very grateful.
Today, I'm grateful as well to you.
Thank you. Today, for the next 20 minutes or so, I will give you a kind of sort of introduction for the project in our lab. And also gonna highlight two projects as examples for the application of the nanotechnology, nanodrugs we developed. We call theranostic, one for the protection of the islet graft and the other one for the nanodrug delivery to the endogenous beta cells. Yes, yeah, let's get started. Here's the, yeah, this is my lab. I mean, we are from Michigan State University. This photo was taken last July. Two students already left for the medical school. I have new members join us. We need to take new photos, I know. Before I talk about the science in my lab, I want to start my discussion with this famous papers published in 2000 by Dr. James Shapiro. I still remember the first time I read this paper. I was just finished my radiology residency and about to start my MD PhD training in Shanghai. And I'm learning the interventional radiology procedures, and I saw this paper for the islet transplantation, and especially I figured out for the cell transplantation, actually, the interventional radiologist, they used this procedure called transhepatic portal vein catheterization for the cell transplantation. I was so fascinated. I was like, I have to do this for my PhD project. So
that's great. The paper could really spark that, much interest and really drive you into the field. I think that's fantastic.
Yeah. Yes, that's true. That's the beginning of my journey for the islet transplantation. back to the paper published 2000, everybody is so excited because of promising data Dr. Shapiro presented. Unfortunately, if you look at the second follow-up paper in 2006, the result was not as promising as the first one. Almost 80% of the patient who received islet grafts back to the insulin injection, which is kind of depressing. And from this very great review paper summarized all the factors contribute to the islet graft loss. You can tell even before the transplantation, during the isolation, during the transplantation and early post-transplantation, even the immunosuppressant has toxicity on the islet graft. So that all contribute to islet loss for the transplantation patient. That's why, I mean, I think that's a beginning for why we decided to work in this field, trying to solve this problem from the three aspects, as I summarized here. Firstly, we want to track the transplanted cells, and also want to imaging the endogenous beta cells and detect the inflammation and the rejections after transplantation, and also for the type 1 diabetes processing. Second part, we trying to protect these cells from all these damages, and also protect the cells, not just the islet graft, also the endogenous beta cells from the autoimmune attack. The third aspect we have been trying to develop is to provide a new source for the islet beta cells transplantation. Okay, first part, yeah, we trying to use imaging as a tool for the, I mean, tracking the islet graft cells during that time, like 10 years, 15 years ago. It's also the beginning of the molecular imaging. So we trying to combine these two technology, molecular imaging and islet transplantation. Firstly, we labeled islet with the nanoparticles, dextran-coated iron oxide nanoparticles. The reason we chose this nanoparticle because it was approved by FDA for liver imaging and also for the anemia treatment. So we label the cells in vitro. And if you look at the iron staining, you can see the iron nanoparticles here show the like blue dots uptaken by the islet cells in the cytoplasm. Then we transplanted these labeled cells to the animal model. We tested both the small animal model in mice and also large animal model, non-human primates for both intra-portal vein transplantation to liver and also under kidney capsule animal models. You can see the labeled cells transplanted in liver, scattered in the liver. I mean, here you can see the black dots spread in liver, and under the kidney capsule models, the graft looks like a pocket of signal loss under the kidney capsule. And we use this imaging tool. We can monitor the graft loss in different animal models. Here we showed in a non-human primates animal model, we treat the animal with toxin, I mean STZ, streptozotocin, which is a toxin for the beta cells. After the treatment, we can see right after the treatment, the signal of the islet graft from MRI decrease. But if you look at the blood glucose level, it will take like several weeks, 2 to 3 weeks. You start to see the elevation of the blood glucose. So we think that's a good, I mean, window for the clinic. If there's like islet graft loss happens, you can rescue the cells. And we also tested these nanoparticles for the endogenous beta cells imaging. We all know in the islet, there's like alpha cells, beta cells, delta cells, and other cell types. We are mostly interested in beta cells. For the beta cells, we all know glucagon-like peptide 1 receptors is highly expressed on the beta cells. So we conjugated the peptide X94, which is approved by FDA for the type 2 diabetes. So we conjugate this peptide on the nanoparticles, and we firstly tested the selectivity of these targeted nanoparticles in isolated islets. And also we did the double staining of the nanoparticles uptaken and the staining of the glucagon like peptide 1 receptor, and we proved that the receptor assisted endocytosis of these nanoparticles. And we tested these nanoparticles in a different animal model, including the healthy and also type 1 diabetes, and monitored the signals with the 3D MPI. You can see with the development of the diabetes, here's the NOD mice, the signal decrease, which are consistent with the beta cells mass in the NOD mice. And the histology shows the very good selectivity of these nanoparticles injected systemically. Yeah, that's the first part of the imaging part, to see the islet cells and both the islet graft and the endogenous cells. So next step, actually very close based on the first step. We reasoned that because we can label the cells in vitro, and we can conjugate like different moieties, like targeting moieties and fluorescent dyes and other probes on the surface of the nanoparticles. Why don't we try conjugate the therapeutics? So the first idea we had is use siRNA. I mean, it's a small RNA molecules, 21, 22 base pairs. So we firstly we tested 2 siRNA. One's caspase-3, which is major player for the cell apoptosis, and the second one is for the beta-2 microglobulin. It's a light chain of the MHC class 1 molecule and related to the immune rejection and T cell CD8 T cells recognition. So for the caspase-3, I mean, models, we transplanted protected and unprotected cells, islet cells, I mean, to the different side, both side of the, I mean, on the kidney capsule, and follow the graft volume, follow up, I mean, check the graft volume with MRI. You can see from MRI data, the protected group has relatively higher volume, graft volume, which resist in a higher production of insulin and also lower expression of the caspase-3. And we also checked the cell apoptosis, and the protected group has significantly lower cell apoptosis. And for the beta-2 microglobulin, here we use the adaptive transfer model. So if you can look at the the diabetes development, I mean, days, the protected groups significantly delayed in the diabetes redevelopment for about 24 days. And the histology shows the downregulation of the beta-2 microglobulin and also significantly lower infiltration of the CD8 T cells. And for the translational purpose, we also tested this caspase-3 siRNA nanoparticles in a non-human primate study. Similarly, we induced the diabetes for the recipient and isolate the islet from the donor and label the cells with the caspase-3 conjugated nanoparticles, and followed after transplantation, we follow up with the MRI and follow up for the animal for 90 days. I want to emphasize that it is rarely accepted like a 10K IEQ per kilogram. It's generally accepted as the minimum amount of the islets required for the insulin independence for the non-human primates. So for this study, we want to show the protection effect of the iron oxide nanoparticles. We intentionally transplanted the marginal number. You can see the all the non-human primates are transplanted with less than 10K IEQ per kilogram of islets. And if you look at the follow-up data here, I mean, on the right side is the control animal, left side is for experimental animal. You can see, I mean, the blue lights here shows the insulin dose, and we demonstrate very good protection effect of the caspase-3 nanoparticles. And these nanoparticles actually this is collaboration with the Columbia University. So we kind of blinded on the nanoparticles, and this result very convincing.
We just had one question from Jean-Luc Tran. Is this human islets donor?
Oh, those are the I mean from the baboons. It's
non-human primate.
Not primates, not xenograft. It's I mean from the non-human primates.
Mm-hmm. Thanks for clarifying that to the question.
Thank you. And for the protection for endogenous beta cells, we did endogenous beta cells microRNAs microarray, and from this heat map, I mean we take the islet and also the microRNA from islets from the different age of NOD mice, 3 weeks, 8 weeks, and 18 weeks. And we identified a bunch of the microRNAs, and one microRNA draw our attention because that's the only microRNAs increased over time during the development of NOD type 1 diabetes, which is MIR216A. Mm. We did the in situ hybridization. If you look at the second row here, in situ hybridization for the verification of the increasing of this microRNA, and also we tested the one of the target of this microRNA P10, and the expression of P10 significantly decreased with the increase of the microRNA. And at the beginning, we didn't know which microRNA gonna have effect on the endogenous beta cells. So we include both mimics and inhibitors and 2 controls. We injected these nanoparticles through the pancreatic duct injection because there's equivalent, I mean, clinical procedure called ERCP. We use this approach for the, I mean, inject the probes to the pancreatic duct and also bile duct. So we adapt this procedure for the selectivity of nanoparticle, because everybody know if you inject the nanoparticles systemically, a huge amount of nanoparticle gonna end up in the liver and kidney. So we trying to avoid that. Here's the animal, I mean, injection pictures. After the injection, you can see the pancreas tend the color change to the dark green. And if you look at the IPGTT result, actually mimics group showed better, I mean, tolerance, glucose tolerance results compared with the other 2 control and also the inhibitors. And we checked the histology. Interestingly, we found in the mimics group, we found the cell proliferation Ki67 markers highly expressed. We believe these microRNAs are closely related to cell proliferation because it is well known. I mean, in adult beta cells not gonna have the ability, much ability for the proliferation. And we published our data 2 years ago, and last year one group from Canada, Dr. Tim Kiefer group, they also targeted this microRNA. They used the different animal model transgenic, they delay deletion of this microRNA and they shows reduce of beta cells mass, and their results are very much in line with ours.
Yeah, in fact, we had Adam Ramsey for Ask the Expert. He was a student there in the Kiefer lab.
Great, yeah. Would like to have more discussion on that. The last part I want to mention it's still ongoing.
Can I ask a quick question before it goes on?
Yeah.
Hi, Ping, it's Eric.
Oh, Eric, how are you?
Hey, sorry, I was trying to type, but it was too long a question. I think it's remarkable that you showed in those primates that the effect with the microRNA nanoparticle lasted so long. Was it, you said 18 weeks or so, at least?
We you mean the non-human primates model?
Yeah, sorry, the non-human primates, yeah.
Yeah, we followed up for 90 days.
90 days, okay. My question is the stability of the nanoparticle and the microRNA inside the cells and presumably inside endosomes or lysosomes, right? So if you label cells, they end up in these subcellular compartments, and they're acidic and particles degrade, and how do you sort of explain this long and quite remarkable positive effect of the nanoparticles given that?
Yep, that's a very good point. Because the labeled cells, labeled nanoparticles has to go through the endosome escape. Yeah, so we believe, we agree, we believe the protection from siRNA or microRNA are gonna be like a transient, probably gonna last, because if you imagine nanoparticles end up in the endosome and released from the surface of nanoparticles, that's not gonna last long. We believe probably gonna last for 1 or 2 weeks. But the effect we observed, we believe, because for the transplantation model of transplantation, therapy, biggest loss of graft happens in the first 2 weeks. So from our data shows, in the first 2 weeks, actually, around 60% of the graft lost. It's consistent with the histology studies. We believe because the isolation and the eyelids, lack of the blood supply and apoptosis, that's a most difficult time for the eyelid graft. After the 2 weeks, actually, if you look at the signal, gonna be very stable for a long time. Our studies, with the non-human primates, we followed up for, I remember, 180 days. After first month, actually the signal is very stable. So we believe the protection happens in the early stage.
Interesting, thanks.
Thank you for the question. For the last part, we also tried trying to produce more eyelid resource. I'm sorry. Yep, we use the IPS because in clinic, actually you can reprogramming the cells from type 1 diabetes, and for this approach you can solve the shortage of the grafts, donor, and also if you can get the cells from patients, you can also solve the immune rejection problem. And we adapt differentiation protocol for 21 days, and after the 5 stages, the cells start to 30% of the cells start to produce insulin, and 10 to 15% of cells express alpha cell markers, and some of them express both. They were also tested at the RNA levels.
Hi, Ken. I was having my microphone issues. I was trying to ask a couple questions regarding the microRNA effects. Can I ask that question now?
Yes, please.
Okay. So regarding the microRNA, may have potential in inducing pancreatic beta cell proliferation. do you check the microRNA level in the normal versus diabetes islets? This is only number one. Is it only in the islet, or is it systemically as well?
I'm not quite understand your question. You mean did we test the microRNA?
No. Did you look at the microRNA level in a normal versus diabetes islet?
I can went back for this for the endogenous. I think you asked for this one. So if you look at the animal model study, we used 3 weeks NOD, 8 weeks NOD, and 18 weeks NOD for the microRNA microarray data. Actually, we believe probably that's your questions. We believe 3 weeks still we consider is normal. Not studying the insulin titers. That's a control. You can see the difference of the microRNAs among these 3 groups. Did that answer your question?
Yeah. So what is red and green? Red meaning less or meaning more?
Green means high. Red
Okay.
means
Okay.
I'm sorry. Red means high. Green means low. You can see
okay. All right. So as your diabetes progress, you get more microRNA.
Yeah. Not all of them. We identified I remember around 200 microRNAs expressed significantly different among these 3 ages.
So this is the islet data, right?
Yeah, isolated islets.
Okay. So what is your opinion? Is it the microRNA being produced from the islet or produced somewhere else?
I think all cells express has microRNAs in the cytoplasm.
Okay. So as as diabetes, I'm sorry, ladies and gentlemen who participate here. I'm just very curious, so I'm trying to participate and try to understand a little bit here. That as the diabetes progress, right, more beta cell is dying.
Yes.
Right. So the ratio of the number of beta cell is constantly decline, i.e. reduced, but at the same time you see an increased level of microRNA. Is that suggesting the microRNA being produced by other cell rather than beta cell?
Interesting question.
From our approach, we only isolate islets. We didn't isolate the beta cells. So I don't think I can answer that question. That's a very good question. Yeah, if we can isolate the beta cell, we tried because the isolation procedure can affect the cell function viability. We feel probably that's gonna also affect the microRNA expression levels, gonna add more confounding artifacts. So that's why we decided to only isolate the islets.
Yeah, and thank you. I understand that this is in the exploration study phase. A lot of things unknown, but thank you for answering my question. That's okay.
Thank you.
Sorry to hold you back.
No problem. It's just for discussion. So I'm gonna try to finish this quickly. So firstly, we label the cells similarly as I mentioned earlier. So because we got a new imaging modality, magnetic particle imaging, it's different from MRI. MRI is more like indirect. Because of contrast, indirect imaging modality, but for the magnetic particle, you can detect the iron oxide nanoparticles directly for better quantification. So we also developed machine learning algorithm for the quantification. You can see here video for the different sections from MPI. But look at the data. We started this project like three years ago. Look at the data we found. There's a problem. Follow up the signal of the transplanted islet organoids actually decrease. We feel even even more significant than islets. We thought probably there's a problem for the vascularization of the islet organoids, because look back at the differentiation protocol. So I mean, the cells IPS started differentiate towards the endoderm at the stage 1. So there's no I mean, chance for the cells develop vasculature in islet organoids. So we believe that's probably the problem. And here also include the one paper published last year, also by Dr. James Shapiro. So he made another milestone contribution for this field for the stem cell transplantation. Their approach is trying to use micro encapsulations. And if you look at the pictures, there's a holes on the encapsulations, and the I don't know, the vasculature gonna like a grows into these packed islet organoids or beta cells, differentiated from the IPS.
I can make a comment to that if you want. So I tested that previously. But I run the encapsulated islet transplant from the stem cell differentiation for 2 years. So what I can confirm for you for sure here is that after 3 months beyond, when you take the capsule out, you see a very rich vascularization surrounding that encapsulated. It's like a tea bag, but it's very small tea bag. Yeah. So and that associate with normalized blood glucose of diabetes animal model, right? So without transplant, the animal have glucose about 400, 500, 600 milligram per deciliter. So after 3-month transplant or so, the animal is surviving on the normal glucose level, about 100, 120. Basically, that's human glucose level. And that indicated that due to the facts that the iPSC is fully differentiating into pancreatic islet, and it producing insulin corresponding to super hyperglycemic mouse model. And then it continued to produce to the point where it sustained the normal glucose. So this is something I actually have firsthand experience. Actually, I run that project for 2 years.
Great. Yeah. Have you here add more details? So we our group, we trying to solve this problem with a different approach. We use 3D bioprinting. As you can see, we designed this we call the super islets, and also differentiate IPS to the endothelial cells. And here I have yeah. Here we have video for the printing of these constructs, and we already got some like structures, but we still have problem for the, if you look at the grades, because the resolution here, we still have problem. But the good news, we're gonna have a new 3D printer very soon, and the resolution gonna be very much improved from 400, 300 micron to the new one to 2 micron. So hopefully we can solve this problem quickly. To summary, I just showed you, because the limited time, I just showed you several projects we work on with the nanotechnologies, and there are other projects like immunotherapy, and also we trying to combine RNA lipid nanoparticle with our iron oxide nanoparticles. There's so many I mean applications we can work on, and I would like to thank all the lab members to contribute to all these projects, and my mentors and the collaborators and the funding. Thank you.
Thank you so much, Dr. Wang. This is a really interesting series of observations and discoveries. I wondered if, it looks like Dr. Tran is here. I believe I think he's the CEO or CSO of Leo. And are you open? Is your group open to collaborative efforts and, research with others who are in industry or other, institutions that you might be able to form some alliances to move the ball forward quickly?
Yeah. So Leo is not specialized in diabetes. Leo only focus on dermatology. So I'm really interested in this because mainly because I work on diabetes for 20 years.
I see.
Yeah, I work on early, mid, late stage type 2 diabetes. So and I find the transplantation is uniquely beneficial for not only type 1 diabetes but for late type 2 diabetes as well, because right now that people, when they're diagnosed with type 2 diabetes, they put on therapy, but over time, maybe 5 years or so, they need to be on insulin, regardless of what kind of therapy they're on, due to the fact that relating to pink presentation that beta cell continue to die. Right? There is no really fully well established reagent now that is able to regenerate beta cell.
Yeah, and
meaning that there's a lot of unmet need that to be able to find mechanism or target that allow beta cell regeneration endogenously.
yeah,
At the moment, there's no such thing, right? You're talking about exenatide, you saw metformin and sitagliptin and P4. They don't do that.
Yeah. So that market is a big market, and I think but there's definitely still and that there's a lot of people delving into that right now. But I think that the type 1 diabetes business case still remains pretty strong because there's a huge health burden that the government and hospitals are paying for when pediatrics patients come in and are diagnosed early in life, and they have to carry that burden for the rest of their life as a type 1 diabetic. So I think both, there's great cases for really pushing into this frontier for both type 1 and type 2. And I think that's, there's a lot of work to be done. I mean, Vertex is way out in front, I think. Viasite is there, and others trying to really crack that the code so that you can have a real viable implant. And I appreciate so much, Dr. Wang, all the efforts you've put forth into this realm. And I hope that, in terms of, we're kind of constrained by time for Dr. Wang's schedule, but hopefully that people who are on the call can reach out to him and speak to him, more at length. So I'm just going to say thank you so much, Dr. Wang. I really appreciated your really interesting work, and I look forward to seeing what comes out of your laboratory next.
Thank you so much.